Thanks for the introduction, Ben. Again, my name is Kyle Sullivan. I am a computational systems biologist working with Dan Jacobson's team at Oak Ridge National Laboratory. And I'm excited to share our work today on two KBase apps that we've developed that use the network algorithm random walk with restart and high performance computing or supercomputing derived networks that we're using to explore plant gene linkages. This has been a team effort between several national labs, including Oak Ridge, Lawrence Berkeley, and Brookhaven National Labs. And we're excited to go through these apps with you today. So should mention that we have a manuscript in prep here. These are a list of the team members that have been involved in this effort, and we have a DOI for the software that's underlying these KBase applications. And so today what I'm going to do is go over an example use case for exploring plant gene linkages in the narrative that I'm sharing now, where we've ran a simulated genome-wide association study, or GWAS, to discover the genetic variants and associated genes and pathways related to a given trait of interest. But before we go through this narrative and show you how you can use the RWRtools apps for your research, I wanted to start by just going over the fundamental reason why we've developed these apps. And the reason is that there's many times in a research project where you run an experiment and you're left interpreting a list of genes as output and how these genes are biologically relevant. For example, you could be applying a treatment to a biological sample. You may want to identify differentially expressed genes between your experimental and control groups using RNA-seq. And then you want to interpret what are those differentially expressed genes doing in a biological context. Same concept also applies to things like proteomics, metabolomics, et cetera. And then another use case, which we'll talk about today, would be GWAS, and in which you're trying to identify what are the genetic variants and associated genes associated with the trait of interest. So regardless of which use case you're starting from, sometimes it's not immediately apparent how all of your genes are related to each other. And so the first place people would typically go would be running a gene set enrichment analysis with things such as gene ontology or GO. And we'll discuss how you can still factor in gene ontology enrichment into your analysis pipeline with RWRtools. But first, let's talk about random walk with restart and our example use case in pipeline for using RWRtools with plant species. And so the two RWRtools apps that we've developed are called RWRtools CV for cross-validation and RWRtools LOE lines of evidence. And so we're using the network algorithm random walk with restart, which, as we explained here, is a network traversal algorithm to explore networks in the model plant species Arabidopsis thaliana. But I've got this text here, but what I want to do for the purposes of this demo is switch over to some slides to illustrate what RWR is and what the output of random walk with restart is as well. And so what random walk with restart is it's exploring a set of connections, and this algorithm uses the same principles that the algorithm PageRank uses to rank websites when you input a query term into a search engine such as Google or Bing, etc. But here, instead of inputting text and exploring connections between websites, which you would see with something like a search engine, in these apps, we're inputting a list of starting genes that we've derived from our experiment, and then in identifying which genes outside of our starting gene set are explored most frequently based upon the idea of guilt by association, that if genes are highly interconnected, they should be part of the same unified pathway. And so you can see that we would start from our seed from a list of genes and individual genes of interest from our starting experimental gene set, and then begin to explore networks of gene-to-gene connections based upon different types of biological evidence. And so one way to explore would be to simply look at what are the nearest genes connected to a single gene of interest from your starting gene set. However, what this is not going to do is not going to efficiently explore all of your different network connections. And so the next expansion upon that would be to do a random walk where you start from each of your different genes of interest from your experiment and begin to randomly explore a surrounding gene-to-gene connection. And so what this would do is would explore a bunch of connections within your network. But the caveat being is that if the random walk would get stuck in a region of low connectivity, you could spend a disproportionate amount of time exploring back and forth between parts of your networks that have lower connectivity. And so random walk with restart expands upon this by having a restart parameter in which at any given decision point, you can either explore a surrounding gene-to-gene network connection by your type of evidence, or restart at one of your seed genes of interest based upon this restart parameter. And so this allows you to efficiently explore your networks thoroughly based upon different types of evidence, and also preventing you from getting stuck in local loops, such as I mentioned here. And so in addition to using random walk with restart, we're also using multiplex networks in which we're keeping different types of gene-to-gene connections from different types of evidence in separate types of connectivity, in which based upon the individual type of biological evidence, those gene-to-gene connections are separated, and then at any given point, the random walker can either choose to stay within a given type of biological evidence or transition to a different type of biological evidence and start to explore that set of network to gene-to-gene connections. And the reason we do this is that previous groups have demonstrated that doing this in this multiplex effort, in which you separate these specific types of biological evidence, gives you more biologically meaningful results. And so now let's talk about what network layers we have within RWRtools apps and what multiplex networks are available within KBase. And so here we have on the left a description of each of the different pieces of biological evidence and individual network layers. The three at the top were, that we've highlighted here, we've generated using supercomputing, using the Oak Ridge Leadership Computing Facility. And so these are network layers that typically would be very difficult or take an exceedingly long amount of time for a user to use without supercomputing capacity. And so what we've done is we've generated these network layers using the supercomputers at Oak Ridge, and then packaged these individual network layers into a multiplex. The reason is that typically building these multiplex networks itself is one of the most time-consuming parts of the process. And then for these networks that were derived using supercomputing, these typically included either petascale or exascale level complexity. And so these are networks that were derived that required an exceeding amount of computational power. And so by putting these into KBase, we are hoping that this can allow other users who do not typically have supercomputing expertise or background to explore these biological networks as well. And so the first one that we've got here is we've listed as IRF predictive expression. What this is using is an algorithm called iterative random forest, which is a machine learning algorithm to loop through an RNA-seq data set. In this case, this is a publicly available RNA-seq data set from Arabidopsis leaf. And what it's using is using the expression values of several different transcripts to predict the value of a left-out transcript. And what this does is, we've previously shown, gives you an order of magnitude more informative connections using this machine learning algorithm than traditional gene-to-gene co-expression networks. We also have, similarly to this, using that same IRF loop algorithm, gene-to-gene connections based upon CG methylation state. And so this is looking at CG methylation near a given gene, and if two genes have similar CG methylation states that predict one another, then they're connected by this network edge. We also have cases in which we have SNP coevolution networks using an algorithm called DUO. And using Duo, we've identified if there are two SNPs that co-evolve together, connecting using combinatorics to identify which SNPs are typically connected to one another, and we've associated those SNPs to genes to derive gene-to-gene connections. And so these are our three high-performance or supercomputing derived networks here. In addition to that, we've curated several networks from other experimental data sources. And I'll show later where we have all of this documented so that you don't have to simply reference this webinar. We have all of the data sources that we've used documented within the app. So fear not, if we can go back later to illustrate where these data sets are coming from. For knockout similarity, this is from a data set in which the scientists knocked out one gene and measured the phenotypic effect on knocking out that gene in Arabidopsis, and did several of these knockout experiments in parallel. And so what this does is if gene A was knocked out and you see a resulting phenotype, and then you were to knock out gene B and see that resulting phenotype, if those two phenotypes share a semantic similarity to one another, then we connect gene A and gene B. So for example, if knocking out gene A was annotated as reduced plant height, and knocking out gene B was a phenotype of plant height reduced by 2 centimeters, for an example, then those two, gene A and gene B, would be connected to one another because the resulting phenotypes from knocking out each gene in isolation were semantically similar, and so we connect them together in this network layer. We also have two different transcription factor target networks. One of them is called regulatory empirical. This is from a data set in which they've done rigorous validation to see which transcription factors have effects on expression levels of target genes, and then regulatory predicted is doing a similar process in which they're using computationally derived associations to associate transcription factors to target genes. Protein-protein interactions. In this network layer, we're connecting up two genes if the proteins encoded by those genes share a physical association with one another. And so these are derived from experiments such as chromatin, or such as immunoprecipitation, and other associated protein-protein assays. And so these are highly curated, experimentally derived protein-protein interactions. This next network layer, metabolic reactions, is looking at whether two genes are annotated to being in the same metabolic reaction pathway. And so if the two genes are part of the same biological reaction pathway, then they're connected by gene-to-gene connections in this pathway. The next network layer is gene ontology similarity. This is similar to the knockout similarity network, in which we're looking at the associations from individual genes with gene ontology terms. And so if gene A is connected to a gene ontology term such as transcription, and gene B is also connected to the gene ontology term transcription, then we link up genes A and B based on the fact that they are related to similar Go terms. The codomain network is based upon whether the proteins encoded by the same genes share a similar functional domain. And so if two genes share a similar functional domain, we connect them up via the codomain network. The final network layer that we're using is a gene atlas co-expression. And so this is from a gene atlas data set from Arabidopsis, in which if two genes are co-expressed based upon correlation metrics, then we connect them up together. And this is sort of a complement to what we have in the IRF predictive expression network. Okay, so now that we've described the individual types of biological evidence contained within these individual network layers, let's talk about how we've packaged these together into a multiplex network, such as we've tried to illustrate on the right here, that we've pre-constructed for KBase users. And as I mentioned earlier, the reason that we're pre-constructing these is because typically the multiplex construction is one of the most time-consuming parts of this process. And so what we've tried to do is try and anticipate what's some of the most common use cases would be for KBase users, and keep these available within our RWRtools apps, so that everybody can use them in their narrative. And so starting with the default multiplex here, we've called this the comprehensive network. The comprehensive multiplex has nearly all of the layers that we have generated, including the 3 petascale or exascale networks. And so we've included this with the idea that users who are trying to gain functional context of their gene set may want to use as many different types of biological evidence as possible to rank the associated genes with their starting gene set, and from there can start to explore the greatest number of network associations. The second multiplex we're calling close to phenotype. This contains the knockout similarity network, as well as the protein-protein interactions and metabolic reactions. And our idea behind this is that if you've got a given phenotype, that starting from the protein-protein interactions, metabolic reactions, and knockout similarity may give you a good idea of some of the gene-to-gene connections based upon these types of evidence. We also have predicted regulation, which includes the predictive expression and predictive CG methylation networks, as well as gene atlas co-expression and predicted transcription factor-target relationships. And so this is intended to contain a large amount of in silico derived predictive associations for your given gene set. In contrast, we also have here what we're calling the high confidence multiplex network. And the idea behind this multiplex is that we thought some KBase users may only want to include network relationships that are derived from highly curated experimental evidence. And so in this one, we've included the knockout similarity network, protein-protein interactions, and the empirical and predicted transcription factor target networks. We also have expression regulation multiplex containing the gene atlas co-expression, the empirical regulatory and predictive expression network. And this is intended to look at various transcription factor target relationships and resulting effects on expression. We have an entire network just dedicated to gene ontology similarity. And similarly to that, we have this gene ontology similarity packaged with the knockout similarity network to create what we call the ontology multiplex. We also have a network only including the metabolic reaction network. And then finally, we have a multiplex that we're calling unknown function. We designed this multiplex with the use case in mind. If someone has a gene set containing genes or proteins of unknown function or have very little annotation associated with them, and you'd like to glean additional functional context for your user gene set, if you're not getting what you if you're not getting as much context as you would like from the comprehensive network that we have, in addition, the codomain network packaged with the protein-protein interactions, metabolic reactions, and dual coevolution to try and explore some of the perhaps lower annotated gene-to-gene connections, particularly for proteins of unknown function. And so now that we've described random walk with restart and our individual network layers and multiplex networks, I'll briefly talk about our use case and then bring us back to our example KBase narrative. And so in our example KBase narrative, we simulated a genome-wide association study of a trait called well-watered shoot biomass in the bioenergy feedstock Panicum virgatum, also known as switchgrass, to look at the genetic architecture of shoot biomass and then understand what are some of the related genes and pathways. So in the narrative, what we do is we've simulated a GWAS assigned single nucleotide polymorphisms or SNPs associated with our GWAS trait. to nearest switchgrass genes in the genome. And then we've used phytosome from the Joint Genome Institute to use comparative genomics to assign the switchgrass genes to orthologous genes in Arabidopsis thaliana. And then from there, we've used these Arabidopsis orthologs as starting seed genes for random walk with restart exploration among our Arabidopsis gene-to-gene networks in our 2 KBase apps. And so here we've identified from our switchgrass genes, we've included 80 Arabidopsis orthologs as our starting gene set in the KBase narrative. So now let's head back to the narrative and see how we can deploy this use case. And so starting from the overview here, this is describing a lot of what I just showed in the slides, but I'll also show you here, I mentioned that if you wanted to see where some of the networks came from, we have the details of that at this link here, which brings up this page. Here we have descriptions that I just showed you about each of the multiplex networks that are in the app. And so within each multiplex, you can see the different types of evidence within that multiplex. And then also, if you want more descriptions about each individual type of evidence within the networks, we have those descriptions here, and we have the DOIs listed for each experimental data source for each of these network layers. And so also I should mention, if you have any further questions that you think of later, feel free to email me or reach out over the KBase users Slack workspace, and I'm happy to answer any questions you may have. And then, as Ben mentioned, if there's time at the end, we're also happy to keep fielding questions there. And so here's an overview of the narrative. First, what we're going to do is choose the genome of interest and start to build our gene set. And so the first thing that we've done is we've imported the Arabidopsis thaliana genome, which you can do by clicking here and going to, if you go into public, you can search for NCBI genomes or phytosome genomes, and then start typing in Arabidopsis, and you will see in a moment. You can then pull up Arabidopsis thaliana either here from this assembly, TER 10 or Airport 11. But again, any NCBI or phytosome genome you can import here. And so then what we've done first is to build our feature set from the Arabidopsis genome. So this is just the build feature set from genome. And here what I've done is just pasted in all of the Arabidopsis feature IDs here. And we stored that as a feature set, which we've called shoot biomass Arabidopsis demo. And then what we do is that we take this feature set that we've built and input it into our RWRtools apps. And so the first one that we're going to talk about today is RWRtools CV. And so the purpose of RWRtools CV, which stands for RWRtools cross-validation, is to really find the interconnectivity of a user's gene set. And so let's explore what the app looks like and describe some of the parameters that we have. So first thing here, we have seed gene keys. You can also click on this parameter to get a description of what each parameter is. Here we're including that feature set that we've built about our shoot biomass gene set as the input. And then next we can choose our multiplex here that we've described. And so we can see here that the comprehensive network is our default multiplex. The next thing that we want to know is, or that we want to list, is the maximum node rank. And so this is basically saying, if random walk with restart gives you an output of what genes are most frequently explored, what are the top ranked number? What are the number of genes outside of our gene set that we want to include as ranks? So the default number of genes to show outside of our starting gene set is 10 genes. For the sake of this demo, I've expanded this up to the top 50 genes just to show you an example of how we can expand this. If you want to include a description, that option is here. And then in order to determine the interconnectivity, what we're doing is cross-validation by splitting our gene set and then using the starting genes to rank some genes that were left out. And so the default method of cross-validation here is k-fold cross-validation, in which you're essentially taking 5 folds in which you start with 80% of your gene set and use those 80% of genes to rank the remaining 20% of genes that were left out, and then repeats this in these 5 chunks, and being that the default value of k-fold is 5, in order to try and get at, based upon this cross-validation, how interconnected is your gene set. And we'll get into some of that additional output values for advanced users to start to get at that. So in addition to k-fold cross-validation, we also have leave one out cross-validation, in which all of the genes except for one are used to rank the left out gene. And then we have the converse, which we're calling singletons, in which a single gene is used to rank every other left out gene. In addition to that, for advanced users, we have the ability to change the restart probability of RWR. We've stuck with the default of 0.7 because previous groups have shown good outcomes from different biological use cases of using a restart parameter of 0.7. But you can keep this between 0, which would mean you never restart, or 1, in which you are continually restarting at your gene set. In that case, you would simply get an output of your starting gene set. The tau parameter then is another advanced parameter, which if you have a specific type of biological evidence that you would like to explore more or less than the others, you can change this tau parameter accordingly to spend more time in a different type of biological evidence compared to the others. But our default that we've listed here is listing ones entirely. And what this means is that there's an equal probability of exploring each given layer at any given decision point, that you can either explore a neighboring gene within your given type of biological evidence, transition to a different type of biological evidence layer, or restart at one of your beginning genes in your gene set. And so now that we've described what the different parameters are in CV, I'll also point you to here in the info tab. We have some descriptions about each of the parameters. We also have full documentation here in the app catalog, where we describe each of these parameters for additional reference. And again, another link to where each of the multiplexes and individual networks are. Okay, so now that we've talked about what are some of the inputs and the parameters of RWRtools CV, I'll show you what are the outputs from this app. And so if we go to the result tab, we can see that it generates this network visualization report, as well as some output files, which I'll get to in a moment. But let's go ahead and view what this network visualization looks like. Okay, so this is a visualization in particular led by Dakota Blair at Brookhaven National Lab. And we're really excited to show this to you all because we think this is a really helpful way of trying to explore our gene-to-gene relationships. And so let me orient you first to what we're looking at here. This is a network where the individual starting genes are located here in purple. And so here we can see our Arabidopsis ID for our starting gene set, which we also can see in this bottom table listed under the seed category. So if we click this, you can start to see all of our starting gene sets that we explored with RWR cross-validation. If we now click on one of those genes, that brings that gene to the top of the table. So you can see the gene ID here. If it's not a starting gene, you'll have the RWR rank associated with it. And now if we have gene symbols or other annotations, effects on when you knock it out, or associated Go terms or MapMan terms, you can see all of those labeled here. And then within the network, you can also see those Go terms and MapMan terms that are associated. And so our starting gene set is in purple. And then all of the genes that were ranked highly by random walk with restart are then colored based upon how highly they were ranked. So now if we click on the rank tab, we can see that the top ranked gene is this alpha beta hydrolase superfamily protein. Now, if we select that, we see where it is within the network. We can start to zoom in on it. And so in this case, this was the highest ranked gene. And that is then displaying here in this teal color. And then the lower ranked genes will then be displayed in more of a light blue color. And so now if we select that gene, we now see that this is slightly less teal and closer on this range of a light blue. If you now want to start to collect some of your genes that you're interested in, you can select a gene here and then toggle this in the collected tab. And from there, you can start to collect these different genes together. And now if you want to come back to them later, you now have these collected genes together. If you want to come back and identify, okay, now I'm starting to build some of the context among these genes. So then importantly, in addition to all of the symbols and Go terms, MapMan terms, knockout effects, a key feature of this is then also looking at the different types of biological evidence that are connecting our genes together. And so that's what we're showing over here is all of the different types of biological evidence that are in our multiplex networks that were used to rank the other genes using random walk with restart. And so you can see all of the different network types here. And as I'm hovering over, you can then see a description of the network type and also then a citation of where we got the data from. And so I mentioned before in the networks.md file, we have that data link as well. But if we quickly click on that, you can see that we're bringing you right to the paper where we got that data set in case you're interested in understanding where the source data comes from. And so then the default behavior from the app is to show you just the types of evidence that were included in the multiplex network. And so now we can see here that the knockout similarity is based upon red gene-to-gene connections. Our exascale pen network, our IRF predictive expression is in blue. We have the CG methylation down here in green, the duo coevolution in this lighter blue. And so now we can toggle on and off those different types of biological evidence that we might be interested in. And so again, if the default is we see this open eye, if that type of evidence was included in the original multiplex used for RWR ranking. But if you are interested in network types that were not included, so for example, the gene ontology similarity network, we can also toggle on and off those network relationships as well. So even if those networks weren't used in the random walk with restart ranking, you can display those by toggling on and off that network as well. And so as we're doing this, you can see again that our genes in purple were our starting genes, and then everything else was in this teal to blue color that were highly ranked. And we can see at the bottom here, there are quite a few of our genes that didn't appear to be highly interconnected to one another based upon the different types of biological evidence here. So we'll get to that in a moment of how we then can see these, and maybe we say, okay, maybe there's less evidence to support these genes being relevant to our overall biological gene set. Maybe they're false positives. And so we can now exclude these in a separate feature set and then rerun random walk with restart CV. So now if we go back to the narrative, so that was the report. And so you can also visualize this report in this smaller window you may find that viewing in a separate window by clicking here will give you a little bit more room to work with and explore. And then finally, we have for some more advanced users, some additional files. And so if you want to get the ranks of all of the genes across all of the folds used for cross-validation, we have this full ranks.tsv file, which I'll show here. And so this is just looking at each fold that you've used, the individual network layers that were used, and then the rank of each gene explored with random walk with restart cross-validation for each of these individual folds. And so then you can explore each of the folds. And so for larger gene sets in particular, this file can start to scale into a larger TSV file. We also have then the mean ranks, which is simply getting the average ranks for each gene across each of those folds. And in particular, this in-val set indicates whether or not one of your starting genes is in the top ranked genes based upon the average random walk with restart rank. We also have the summary tab here, which using cross-validation, you can get area under receiver operator characteristic curve and precision recall curve, average precision, and other metrics based upon these different types of folds and cross-validation. We also have the precision and recall of each gene in your gene set for each of your individual, for each of the folds in this metrics rank. And so these are more advanced files that we give out to users in case they want to really start to get at some quantification for how interconnected your genes are. Okay, so now I had said, as we looked in this visualization of RWRtools CV, we saw a number of these genes were less interconnected to the others. And so now in this next part of the narrative, what we've done is say, okay, some of those genes were more interconnected than others, that there were a few of them that were sort of off on their own in the networks. And so we've now built a separate feature set by just looking at these genes and excluding these genes at the bottom that were less interconnected, and built everything else up here that was in purple from our original shoot biomass gene set and put them into RWRCV. And we've called this shoot biomass k-fold filtered. And now looking at the filtered gene set, we can start to see from that network visualization that now we can see, as we load this network, that everything is a lot more connected to one another and that there's very few genes that are sort of off on their own here. And so this is just an example that we're showing to say how you can try and use these the CV app to get at the interconnectivity of your own gene set and possibly use network algorithms to identify, are there genes that might be false positives based on the suggestion that they're less interconnected to one another, and that the underlying hypothesis being that if genes are contributing to the same process, that they should have high network connectivity to one another. And so that is the RWRtools CV app. Now we'll get into the RWRtools LOE app. And the purpose of the LOE app is essentially to get at what is the functional context of a user's gene set. And so in particular, from some of those starting genes, we see that there is a SKS5 oxidoreductase from our simulated GWAS, and then it's connected to a cellular synthase. We've got some descriptions of the types of biological evidence that we're starting to pick up from the RWRtools LOE app. And so quickly, I'll show you what the parameters are for the LOE app. You'll notice that a lot of these look very similar to what we have for RWRtools CV. And so we have our starting genes here, our shoot biomass genes. We'll get to the target gene keys in a moment to describe that use case. But again, we have our multiplex with the comprehensive network being the default, the maximum node rank, which our default is showing 10 genes. Here, we're going to bump this up to 100 and start to really look at more of a broader network. And again, the restart probability and the tau for those advanced users who want to play with exploring the networks and tweak those parameters. And then the output being the top 100 ranks based upon the RWR exploration. And so this is then output in an additional feature set that we can use for downstream KBase apps, which I'll get to in a bit. And so the output then is again a network visualization, which we can view in a separate window. And because we've added 100 genes, I think this is a good indication of you might really want to have that larger visualization. And as we load it up, then again, we have in purple our starting seed genes. And then from here, we have all of the genes ranked from 1 to 100 based upon this sort of color scale of teal being highly ranked and light blue being lower ranked. And so if we start to look at some examples, let's say we have a gene in mind that we want to explore. We can start to say, okay, let's take a cellulose synthase and see if we have any cellulose synthases that are ranked. So we can see here that we found cellulose synthase 6 here. And then we can highlight that within the network and start to see what are some of the surrounding connections. So scroll up. We see that the cellulose synthase gene is up here. Then we can start to explore what are some of the connections with that. And so we can start to also move this gene around if you're starting to see the network gets a little bit too dense to unpack. And actually, if you come back to this report later, this gene will be in that same exact place as you left it. And so if we click on that cellulose synthase gene, we can see that it's connected to one of our starting genes here, which was a cellulose synthase-like D2 gene. And so the D2 gene is connected to this cellulose synthase A6 gene, and that is connected to another starting gene, an oxidoreductase. And we're seeing here that that's connected by one of our predictive expression networks here using petascale and exascale level complexity. And so then you can use this then to start to explore these different types of gene-to-gene connections and understand what are some of the genes that are highly interconnected to your starting gene set, and use this to then start to identify, okay, I have now a alpha beta hydrolase that's highly ranked based upon my starting gene set. Could there be that has an important biological context for my starting gene set? And from that, start to identify some biological pathways that are related based upon network connectivity. And so in addition to the report, we also have for more advanced users, similarly to the RWRCV, we have this ranks.tsv file, which you can get each of the list of genes that were most frequently explored by random walk with restart, and you can save this locally. And so that was using a single gene set to explore others. And now finally, I'll get to this last use case in which, let's say you want to see, starting from your user gene set, how functionally related is your second gene set? And so in this case, we've simulated a GWAS of shoot biomass. Let's say that we've done a separate GWAS of switchgrass height. Can we see if these switchgrass height genes are highly ranked by starting from the shoot biomass genes and using random walk with restart? And so then we build our feature set here of our what we're calling height genes. And then using the RWRtools LOE app, we start by using our shoot biomass genes to explore our multiplex networks. And then we've now included this target gene set, which is our height genes, to see if they are being highly ranked in the multiplex network. And so then if we then look at that result and we load this, we'll see that, again, we have a number of genes that are connected to our starting gene set. And in gray here are our target gene set, which we can see that there's some network connectivity to our original genes, but that we see that a bunch of these genes are not interconnected to one another, and that a lot of the gray genes, in fact, are not connected to our shoot biomass genes, which are in purple, and not connected as well to a lot of the genes that are highly ranked by random walk with restart, starting from our shoot biomass genes, which are on this teal to light blue scale. And so we're using this then to see that based upon network connectivity, our two phenotypes of interest may not be as functionally connected as one might suspect based upon an a priori hypothesis. And so again, we then also have this ranks.tsv file, which you can also see the ranks of that second target gene set in that output. And so finally, then, let's say that we've now gotten this output of RWRtools LOE, where we've said we now wanted to see what the top ranked genes are based upon starting from our gene set of interest, in this case, shoot biomass. Can we then start to do additional downstream interpretation? So in this case, we've taken those top 100 ranked genes from random walk restart and put them in as a feature set object for this app functional enrichment for Go terms in KBase. And what this does then is use your is an example of how you can use the output of RWRtools for other KBase apps that have been available for some time. And so in this case, we're then saying, what are some enrichments in gene ontology terms based upon our top ranked genes from RWRtools? And we can see here in this report then that the top ranked genes based on network exploration include processes like fatty acid biosynthesis, copper ion binding, et cetera. And so essentially anything that would take in a feature set object within KBase could be used. And so basically, this is just one example of an app in which you can use for downstream interpretation in your KBase narrative. And so we're approaching the top of the hour, I see, but we've also finally got some further resources here for the catalog page. We also have a link to the source page for the RWR toolkit, for the RWR toolkit code. But with that, I'm happy to take any questions that anyone might have. If there's something that I haven't yet shown that you are interested in, and appreciate everybody tuning in for the webinar.
Super. Thanks, Kyle. I saw someone starting to type a question into the Q&A doc here. I can also I'll allow everyone to unmute themselves if you just want to do the verbal question. Or if any other KBase folks who are on the line have any additional thoughts.
I guess I have a question, and it's taking me a while to type it, so I wanted to interject. Can you hear me?
Yep.
Yeah.
So what if your list is very noisy? Like contains more noise, and you know that like there are situations that generate such lists, but they might contain more noise than signal.
Mm-hmm. Yeah, that's a great question. So, I mean, I think that's one of the ideas behind the CV app that I was showing here earlier. So, right, in this case, we had a list of genes that we inputted. And then as we started to look at that network, we saw some genes that, whoops, that's the filtered one. We had some genes here that were sort of off on their own island, so to speak, in the network, right? And so that might be an indication based upon the different types of evidence that we're using here to say that maybe they're less connected to one another and based upon the idea that if you've got a noisy gene set, that those noisy genes are probably more likely to be kind of off on their own island, right? Because if you've got a phenotype of interest, the idea is that that the genes that really matter should be connected to each other through relevant biological pathways, and that the things that are more noisy are sort of going to be off on their own and less connected to one another. And so then if you've got a gene set with a lot of noise, I'd suggest starting maybe with the CV app and trying to see and visualize it, with this interactive visualization to see what are some of the connectivity that we're seeing here. Are there specific types of evidence that you want to explore more than others? And from there, can you sort of start to perhaps whittle down some of those genes that are less interconnected? And then from there, input the genes that are more interconnected into something like the LOE app, or simply just increase the number of genes that you're visualizing by the maximum node rank here. And that can give you a starting point for trying to whittle down some genes that you suspect might be noise, but then can still start to visualize what are some of the top ranked genes. Does that answer your question, Laura?
Yep. And I have another question if nobody else has a question, but I'll pause for a moment.
Yeah, go ahead.
So this is super cool. Definitely. I was wondering, so network analysis has been used in Arabidopsis for decades, I guess, and there are tools like String that students would be familiar with going to and utilizing. Have you done any comparison or obviously this is more sophisticated, but
so actually String is actually one of the sources of one of our data sets here. So protein interaction, that's all right. What we've tried to do then is take a similar approach to what String does. String does give you a lot of different types of gene-to-gene evidence, right? And also shows you in that network visualization of what those gene-to-gene connections are based on color coding. And so what we've done here is we've incorporated the protein-protein interactions from String and tried to curate those to the String types of evidence with the highest confidence, and incorporated that along with some other protein-protein interaction network sources into a merged protein-protein interaction network. We've compared them to just looking at String alone, particularly with the protein-protein interactions. And we've shown that merging these different types of evidence together is in some of our testing before we've launched the app, that it was giving us better results. And so that's why we've tried to diversify these different types of evidence. So I certainly think what we have in KBase is complementary to what you would see in String.
We got one question here in the Q&A doc. Someone asks, if we had a region of interest from a QTL study in switchgrass, how would we go about linking genes from these regions for our phenotype?
Yeah, so I think for that question, the first step would be sort of looking through the switchgrass genome and then looking at a given, so QTL is quantitative trait loci, is looking at that region of the genome and sort of within that window, identify what genes are located in that window or windows, and then probably start by including all of those genes as your starting feature gene set. But that could be a case in which you have sort of a noisy gene set, and so might recommend trying the RWRtools CV app to look at, are there some of those lesser interconnected genes, and seeing if that can, if you see some genes in your gene set that are more or less connected to each other.
And one more question here. Are these networks highly optimized for plant genes only, or would it be possible to adapt the same kind of analysis for microorganisms such as fungi?
Yeah, absolutely. That's a great question. In particular, Angie Walker, who's a team member involved in this project, who's since graduated, who's a former grad student. She did some exploration of using yeast networks to explore, which can be used in a similar way that you would essentially try and find a way to transfer orthologous genes from a fungal species perhaps over to yeast or another model organism. And the idea being that for some model organisms, we just simply have more experimental evidence to connect genes together. And so Angie in particular did some work on, which is now published, looking at some yeast networks for microorganisms. But ostensibly this can be used for any species that we have data on, right? And so as a starting point, we've started by incorporating Arabidopsis gene-to-gene networks, but we're looking to expand that as well. We're hoping to include other plant species in KBase. In particular, would be great to include switchgrass or other bioenergy relevant plant species in this, so that users wouldn't have to do that orthology mapping. But yeah, we can ostensibly do this for any species that we have network relationships for. And so that's why we then have the GitHub for users that want to build that on their own. But then the KBase app here for starting with Arabidopsis as our starting point for exploring network relationships. But if you have any additional questions regarding that, please feel free to reach out, and I'm happy to point you in the direction of some of those resources.
Super. Thanks so much, Kyle, for this presentation and this really awesome tool that you all have put together here. Like Kyle said, if you have any questions or you're using this tool and are looking for any insight, feel free to log on to our help board and drop us a question there, or you can reach out to Kyle directly. If you look him up at Oak Ridge National Lab, you'll find out how to contact him there. So thanks everybody once again. We'll post this on the YouTube once it's done recording here, and thank you all for attending, and looking forward to seeing you all on more KBase webinars coming up soon.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.